Our third and last speaker of the session is Bastian. He's about to finish his PhD at the Department of Epileptology at the University Hospital in Bonn in Germany. And a fun fact is that while he's a researcher a day, he's a death metal drummer at night. So that's kind of cool. And he'll be talking about a deep generative modeling approach to detect epileptic lesions.
Yeah, thank you very much for the kind introduction. Let me share my screen here. All right, can you see the presentation? Yes, okay, perfect. So as I said already in the introduction, I'm part of the Department of Epileptology in Bonn, and I would like to present to you today our approach on how we automatically detect very subtle epileptogenic lesions. First of all, for everybody who doesn't know how epilepsy is actually diagnosed, in the pre-surgical evaluation of epilepsy patients is based on 3 pillars. One of them is MRI, obviously. Then we have the EEG and the seizure semiology. So how do seizures manifest in the patient? And last but not least, neuropsychological testing. And unsurprisingly, if you want to look for lesions, MRI is the way to go. It has become most crucial as for this patient, for example. It's a 41-year-old female patient, and she had a lesion. I won't play the guessing game now. She had a lesion in this region here, and our neuroradiologist suspected that it's a so-called focal cortical dysplasia, or in short, FCD. And these FCDs, they are congenital disruptions of neuronal migration or differentiation during brain development. Basically means while the brain is developing, the somata, so the cell bodies of the neurons, are traversing from the neural tube outside to the cortex. And in these patients, they don't migrate far enough, so they get kind of stuck in the deep white matter. It's actually one of the most common pathologies causing drug-resistant focal epilepsy in both children and adults. Drug-resistant epilepsy, meaning that the only therapeutic option for these patients is to undergo resective surgery. And that's exactly what this patient did. So we, or not me personally, but the neurosurgeons cut out this part of the brain. And then we did some histology on it. And we found out, yes, it's actually an FCD, type 2B, doesn't matter what type. And this patient was afterwards seizure-free. So she was healed from her seizures. So you can imagine that positive MR findings are very high therapeutic importance here. In fact, 80% of patients become seizure-free after a complete resection of the lesion. Actually, if you're a very specialized clinic, this number is probably even much higher at like 95% or something like this. However, not all FCDs can be detected easily by the neuroradiologist. This one, for example, was an MR-negative patient in our clinic, and the lesion is here. I marked it green. And this is not a singularity, but in fact, we have only abnormal MR findings in around 50 to 70 FCD patients. This differs a lot on the clinic and on the skill of the neuroradiologist. So there's like a huge number that's or big improvements can still be made in this field. So how else can we detect these subtle FCDs? I mean, it's kind of obvious choice that you try to do some automated classification to help the neuroradiologists. And we are not the first ones to do this. These are 3 of the main publications in the field, I would say, or at least they are my favorite publications. And what they have in common is that they use a surface-based approach to detect this lesion, which makes sense because these lesions are usually at the interface between white and gray matter. And they define some kind of features, mostly like gradients along the surfaces or something like this. And then these features are fed forward into some machine learning algorithm of choice, and then you have your classification. And if you check these approaches, the specificity, at least of the 2 approaches I highlighted here, are very high or actually perfect. But if you check the sensitivity, you can actually see that over the years, the sensitivity of these approaches has not really improved, and it's around 70%, which is kind of the level that neuroradiologists also reach. Some other problems with them is that the human feature engineering itself is a problem because it might introduce some biases. Also, you have to like run the images through some heavy pre-processing pipelines, like, for example, the surface reconstruction, which is always easily flawed. Like there are flaws often, so you have to manually readjust it and stuff. So that's already pretty bad for physicians to do. Also, they're very vulnerable to noisy data, especially motion artifacts, which in epilepsy patients is very, very common. So yeah, okay, we already had 2 talks about deep learning, so the Deus Ex Machina moment is a little bit gone, but this is something that you can do probably much better with convolutional neural networks, and that's exactly what we tried. So what kind of patients did we have? We have 42 patients with histologically proven FCD type 2B. And I want to highlight the number, 42 patients. In deep learning terms, that's ridiculously small. In clinical terms, this is actually quite a lot because it's, yeah, it's not such a common disease. Then, yeah, we have scans. We have T1 images and FLAIR images. And we labeled them, yeah, actually my boss labeled them. He's a physician, he can do it pretty well. Yeah, for validation, we used a 5-fold cross-validation scheme. We calculate the sensitivity according to the co-localization of super threshold clusters. We just had like a fixed threshold of 90% probability with the manual lesion label. And the specificity we tested in an out-of-sample test with 56 age and gender matched patients who also had epileptic seizures, temporal lobe epilepsy. So they also have seizures, they also have some atrophy, but they don't have FCD lesions. So this is really the group you want to distinguish them from. So what do we do? First of all, I'm not an engineer, so I just like apply stuff. I actually used DeepMedic. I'm sure that that Marco knows this this architecture. It's a very famous architecture. It won, I think, the Brett's Challenge in some year. And it's a pretty standard CNN by now, but what's making it kind of special is that it has this dual pathway. Here, the upper pathway where it extracts 3D patches in the high resolution images. So to learn implicitly the local neighborhood relationships. And in the lower pathway, it first subsamples the images, the raw images, and then also extracts 3D patches and thereby learns something about the global neighborhood relationships. Yeah, we just fed in our raw T1 and our raw FLAIR image. And when we do this and just use this off-the-shelf method, basically, we already reached a sensitivity of around 81%, which is pretty nice. But if you look at the specificity, it's rather poor. 16 of the 56 temporal lobe epilepsy control patients also showed false positive clusters. So there's something we need to do. However, we have so few patients, so few data, that we cannot really do a lot of like hyperparameter tuning and stuff like this. So what we thought what we have to do is we have to simplify the problem before we feed our data into the network. And what do? So this is again our index patient. And if you check now the lesion, the area of the lesion, and you know where it is, you can actually see that there's something like a radiographical feature that classifies this lesion, which is an increased cortical thickness and like a blurring of the gray matter, white matter boundary. And this is actually exactly the features that new radiologists are trained to look for. If you now look at the same region, but in the T1 weighted image, you can actually not see that much. So in comparison, again, this is the FLAIR image, and this is the T1 image. So in the T1 image, the lesion seems nearly invisible. And our idea was, okay, hey, can we use this information mismatch between these two sequences? And if yes, how can we maybe quantify this beforehand, before we feed it into our DeepMedic network? And this is where GANs come into play, or at least we came up with GAN, a GAN approach. I'm very grateful that Marco already did a great introduction for GAN, so I won't get too much into detail with this. You heard this already. But just in our case, what did we do? Our real data were the FLAIR images of the patients, which actually do show this, these radiographical features of the lesion. Our source data for this conditional GAN were the T1 images, which do not show this radiographical feature, or nearly not. And the synthetic data that we wanted the GAN, the generator, to produce should be something FLAIR-like, or something that looks like a FLAIR, but that does not show this radiographical feature because it's only based on the T1. And so, yeah, we train this GAN, and then with iteration to iteration, the output of the generator becomes better and better until the, sorry, is it stuck here? Yeah, until the discriminator does not know anymore, is it real or fake data? We have now photorealistic FLAIR images. Here, just just showing off a little bit here. On the left-hand side, there are the real images, and on the right-hand side, you see the synthetic images of our GAN. Our GAN, by the way, was a 2D GAN. It was actually the pix-to-pix that Mark already mentioned. Okay, and if we now look at this for our patient, again, this is the real FLAIR. Here you see the lesion highlighted. And if you now look at the synthetic FLAIR, which is based on the T1, you see that the lesion is invisible here. But apart from that, the intensity histograms are so similar that we can now simply subtract these two images from each other, and then visualize the difference map in colors, in the color scale, for example. And just by visualizing, the FCD region already pops out pretty nicely. Now, what we did in the next step is, of course, we developed this two-step procedure. We trained the scan on, by the way, healthy controls, because we didn't want it to learn anything about the lesion. Then we have the synthetic FLAIR and the real FLAIR. We subtract them, and we then input not only the raw T1 and the raw FLAIR, but also the difference image into DeepMedic, and and train it. And now we come to the results. Our first classifier, you know it already, pretty bad specificity. And now, not a big surprise, our second classifier with a two-step procedure worked much, much better. In fact, it has a sensitivity and specificity of over 90%. Also, the number of extralesional clusters within the patient brain is much less. So it really improves a lot to feed in the scan difference image. And here are some, a little bit cherry-picked results, I have to say. This is the index patient. You see the GAN difference highlights the lesion and the classifier finds it. This is another lesion which is actually visibly already visible. And the GAN difference also finds this one. This one is a patient which I think really highlights the benefit of this of the deep learning approach, because you can see that this image has quite a lot of motion artifacts, very, very common for epilepsy patients. But still, the GAN difference image highlights the lesion, and the classifier also finds it. And last but not least, also one result where our classifier did not find the lesion. Also, the GAN difference is kind of it's mostly noise. But now, okay, my last slide, and actually the most important question about it, does it also generalize to other data that's not our data set? And in fact, it kind of does. These are also small sample sizes, yes, but they are from different scanners and from different clinics, the University Clinic in Frankfurt and the Charité in Berlin. And also here, our sensitivity is in all cases above 80%. Actually, the lowest number is 80%, but I also want to highlight that this here is a pediatric patient cohort. So it's not adults, but it's children brains. And everybody who ever worked with a pediatric patient cohort knows that these brains look very different to adult brains. Yeah, so that's already it. Thanks to all my collaborators. Thanks for your attention. And I'm really happy to take questions now.
Great. Thank you, Bastian. So everyone, could you please put your questions in the Q&A? Oh, they're coming in. So Daniel asks, do you really need the pathway for the global structure in your CNN? The lesions look small and local.
Yeah, very good question. And to be honest, I have not tried to only use the local. Oh, I'm sorry, somebody's ringing my bell. I've not really used a CNN that only uses like one pathway, basically. But I'm pretty sure that for FCDs, it's useful to understand the global neighborhood relationships, because FCDs they can appear everywhere in the cortex, but in some parts of the cortex more often than in other parts. So I think that by having this global pathway, the CNN already learned something about also the general distribution of FCDs. So I think the global pathway is actually helpful.
Cool, thank you. I have a question. So have you looked specifically at your false positives and why they might occur?
Yeah, that's a good question. So false positives are like, so in general, our approach is very sensitive to everything that looks different in the FLAIR than it does in the T1. And therefore, since our control group here was a temporal lobe epilepsy group, it sometimes happens that these patients have hippocampal sclerosis. So the hippocampus itself might also be hyper intense in the FLAIR. And therefore, many of our false positives actually were within the hippocampus. So it actually, our approach kind of found the lesion, but it's just not an FCD lesion. Actually, in an approach that I'm using now, I just like, I know that FCDs will not appear in the hippocampus, they will only appear in the cortex. So I use like search masks only around the cortex. So that already gets rid of quite a lot of false positives.
Awesome. Okay, if there's no other questions.